I suppose when you and I started off originally, would have created new collagen through repair mechanisms that we still have, maybe with lasers, radio frequency, but these are the new kids on the block. So what have we got? We got exosomes that we've seen earlier, and exosomes, I suppose, work within the cell. Exosomes, in many ways, I suppose, are a bit like the COVID vaccine. They've got this lipophilic structure around them. They're within the cell. They're acting at nuclear level. They've got codes and biochemical cues that make, I suppose, gene encoding possible. And as a consequence, the new collagen is formed that way. But polynucleotides are different. Polynucleotides are really just peptides. They're proteins. And what they're doing is going into the cell from the outside. So one is extracellular, the other is intercellular. Extracellular means that they must come up to the door, knock on the cell to get into it. And they are a bit like, I suppose, the COVID virus was, because it could only get into the cell using ACE receptors. Polynucleotides use A2 receptors. So what do they do? Once they're outside the cell, they have this, I suppose, inflammatory reaction that we know that we're trying to suppress to a large extent. And they're made up of these nucleotides, aromatic compounds. They have got active free oxygen residues, and they can be used in many ways to hydrate, and they can also provide protection against UV light and mop up free radical scavengers. They do all these things, but I suppose what we're interested in today is how they work to produce collagen. So we've used them for quite a long time before in medicine with the chronic, I suppose, wounds, burns, that sort of thing. We know they've worked in sort of tissue repair, and they're involved also, as I said, in hydration, scavenging free radicals, stimulate cellular growth, and obviously the anti-inflammatory effects work by modulating cytokine release. And I'll go into all that just later in it. So we have the antioxidative effect, moisturization, looked at to an extent. Now look at them a little closer, but really what I'm interested in, and I spent a lot of my life as well working on this, is how they affect fibroblasts. So I'll just quickly look again at the antioxidant effect. So that is free radical scavengers, and as a consequence, we know that this is used in many ways to protect the skin against things like UV light. That works by a reactive oxygen sequence. If you remember your biochemistry, the donator electrons from one molecule to the other, and as a consequence, stabilize these free radicals from causing cellular damage. So I suppose, DNA protection is one of the big things that we wouldn't really notice when we use them as doctors. They're very, very safe. They're coming from a safe source. And if they were doing nothing else, they provide moisturization. And they do that by this mechanism where hydrophilic polyanic structures enable them to bind water molecules. So they fill up the intradermal spaces by making the tissues firmer and more hydrated. But that isn't, I suppose, really what we're looking at. Even though this produces suppler, plumper skin, which obviously from the patient's point of view will reduce the appearance of fine lines and wrinkles. But what we want to do is more this, the wound healing repair, the effect they're having on cell proliferation, the effect they're having on extracellular matrix. And we know that exosomes and secretomes can work on cell senescence. And cell senescence is going to be a big thing in the future now to stop cells, zombie cells, from dying off, that they can remain there. And as a consequence, people can probably look younger. And we know some of the companies now with secretomes are actually moving into producing drinks, chewing gum, intranasal sprays. So that's going to be the whole new thing. So what we're looking at is how do they affect fibroblasts and how does the collagen produce them? And then I'll go into how we actually use them, the mechanism of action. So fibroblasts originate from one of our previous doctors shown that can be affected by exosomes by working through DNA sequencing directly on the fibroblasts. These guys are a little different. They're working through, they're knocking on the door to get into the cell. They're using this A2 receptor mechanism. So A2 is like the lock and the key that gets you into the cell. And there is a good article there by Squadrito, Francesco, and Beto that showed how this works down through the different cytotomes, the cytokines, the growth factors, and as a consequence, we will get the polynucleotides working on the fibroblasts. So just a quick look at adenosine A2 receptors. What are they? They are involved, obviously, in inflammatory, reducing inflammatory cytokine production. And I think with the delayed onset nodules that we get from a lot of the hyaluronic acid fillers, that this could be a way of almost preventing this in the future. And I intend to start doing some sort of work, I suppose, on this very soon, experimental work. I'm working in medicine less now than I used to be, but this would be a good project for somebody to take up. So it's working through the A2 receptors that will promote collagen production. Now, the polynucleotides will stimulate fibroblasts, as we know, and the fibroblasts are the things that are present in connective tissue. They're motile cells that can move about the body. You can move them to reach red light. And in one of my lectures in India, I think it was, actually, I showed that where we had an electron microscope and we had an obstacle, and as a consequence, the fibroblasts, if you shone light 633 nanometers, the fibroblasts will jump over the obstacle, which I find is fascinating. They're motile sort of cells. They can be moved around the body, but we can also control them where they go. Now, they'll produce the extracellular matrix components, collagen, elastin, glycoproteins, all the things that we need to keep cells healthy. And as a consequence, we use these quite a lot for that particular reason. So they offer a multifaceted approach to skin rejuvenation by, as we said, originally hydrating all the cells, but also boosting collagen production, promoting healing, and because of their origin from the source, they're comparatively safe. And I'll go through that in a moment as well, where we get them from salmon sperm and why we do it. Growth factors are an important thing because growth factors, I suppose, are the most important, facet really in skin repair and regeneration. We've been working with them quite a long time with where we won awards for using stem cells originally to promote growth factors that through PRP also, and we were using them for repairing burns, as you know, and for also tissue damage secondary to vascular necrosis. So this is the way that they would work there. We were looking originally at the growth factors. There's 7 of them that sort of work through the fibroblasts to produce collagen, and also, of course, cytokines, which are very, very important. Cytokines, as you know, work as signaling messengers, and the polynucleotides that we're injecting in will modulate the cytokine production, leading to enhanced healing and, of course, also reduced inflammation. So if you want to get to know these things, you've got to know the physiology of how they're operative and why they're working. And we know that fibroblasts per se can produce collagen. You see it in the right-hand diagram here. They contribute to skin texture, elasticity, firmness, and there's restoring the internal structural integrity of the skin itself. A2 receptors is the code that they sort of work through. And one of the big things, I don't know whether you know about them or not, but they really work through these GPCRs, which is G protein coupled receptors, and A2 really are part of that larger family. So they're involved in transmitting chemical signals across cell membranes. So this whole process of bioregeneration works like this. So as a consequence, I will show you now in terms of administration. This is the level that we have to inject them. So it's in the deep layers of the dermis. They can be injected subcutaneously or intradermally. Obviously, if you run them too high in the skin, you're going to be left with little sort of nodules. And they come from salmon, as I sort of mentioned, and salmon sperm DNA has been used for quite a long time. It's nothing really new in medicine. I know it has crossed into, I suppose, aesthetic medicine in pretty much the same way as Botox did originally. As you know, with Botox, we were using it originally in ophthalmology before it was used in aesthetic medicine. The same thing with PRP. It was used in dentistry and orthopedics.